vaccines and other new formats as they are on the way. So who am I? Dr. Zhao gave an introduction. I grew up in Watertown, Massachusetts, very close to Boston, which explains the incurable disease of being a fan of the Boston Red Sox. We grew up very close to an interesting place, the Helen Keller Perkins Institute for the Blind. And so some of my earliest memories are of people with illness. It was not unusual to see patients from the Institute with neurofibromatosis, which can cause blindness, waiting for the bells which rang on all of our streets to indicate that it was safe to cross. That was an unusual thing about living there. Another early medical encounter occurred just after running into a hornet's nest and getting covered. It was an interesting experience because after running into the road and almost getting run over by a car that looks something like this, that should give you an idea of how old I am. The doctor driving the car proceeded to take off his sport coat, swat off all the hornets, fix me up, and bring me home. Well, I think a light went off or turned on in my head, and it hasn't gone out yet. So after medical school at Navarre, think running of the bulls in Spain, and a stint in Baltimore for internship and residency, as Dr. Zhao pointed out, a couple of years in St. Louis, and then many years in practice in Northern Virginia, and then in June of 19 to Virginia Commonwealth University. Okay, so we're going to have a lot of choices going forward with COVID-19 vaccines. I don't think it'll be that long before we're seeing another, who knows, 5 or 6 vaccines in the next year or two. But we really have to, I think, understand the options that are as they become available to us. So today we'll focus in on the basics of mRNA vaccines, from their construct to some adverse reactions. And I do want to focus a little bit on 3 potential adverse reactions: enhancement, cell signaling with the spike protein, and systemic reactions. And we'll go over all these. It's impossible to go over certainly those that are even in phase 3. We've got 8 or 9 vaccines in phase 3. So it's going to be impossible to go over each individual vaccine. So you're all aware of these issues, that along with hand washing and antibiotics, vaccination represents clearly our most effective weapon in an attempt to prevent and control infectious diseases. We have eradicated many diseases, drastically reduced the incidence of many diseases. And so we're looking at approximately 30 diseases worldwide that can be prevented by vaccination. Well, the first trial really of a vaccine was in 1796 with cowpox. And now 225 years later, we've evolved from the principles of pathogen isolation, inactivation, and injection to a very rational vaccine design based on genetic engineering, immunology, and that's of course our area, and structural and systems biology. The truth is, however, that there are many unmet needs in the field. Certainly pathogens which can evade immune responses or which require both cellular and humoral immune responses. There are so many emerging infectious diseases. Again, we're in the midst of one, but Zika, Ebola, pandemic influenza. And just a side note on this, if any of you are looking for a fun read, as long as you don't mind being kept up at night, you might take a look at David Quantum's book, Spillover, which is quite interesting. So we need a vaccine that enables rapid development. It can be deployed rapidly and is amenable to large-scale production and distribution. So as you are all aware now, mRNA vaccines really meet these needs. But the reality is, even with that, validation of both efficacy and safety must be confirmed in all cases. So our hope is certainly that we're going to get a return to normal human social interaction and life with vaccines. And really, that's probably the only way it's ever going to happen. The safety margin, again, is high. We are administering this to healthy individuals, such as many of our people who are in the medical field. Nonetheless, especially for medications approved with emergency use authorization under EUAs, long-term monitoring of those who receive the vaccine is crucial. It is true that the immune response that most rapidly and dependably clears a virus is the one that we want. We want to understand that, and we want to replicate it as much as possible. However, as you have all seen, we have asymptomatic carriers, and we have people who succumb to the disease very quickly. So immune responses are highly varied against this virus, suggesting that there may not be a single perfect strategy. Let's read into that. There may not be a single perfect vaccine, which is going to solve the problems. We are so fortunate that many vaccine candidates are being developed of all different sorts. And what I envision going forward is that we're probably going to be targeting certain populations of patients with a certain vaccine. One patient population may be amenable or better suited to a certain vaccine and others to another. And I think that's pretty exciting given what we're facing in today's world. Okay, which brings us to nucleic acid-based vaccines, which, as all of you are aware, we identify a target gene, it's sequenced, synthesized, cloned into a plasmid DNA template, transcribed in vitro, and this is crucial because it's going to speak to the safety. They're transcribed in vitro, there's no cell compartment, no cell culture needed. Vaccine is administered and utilizes host machinery for translation into protein. And this will hopefully elicit both a humoral and cellular immune response and generate some immune memory. Now, before we get there, the antigen that we're targeting should be harmless. It should be a harmless piece of the virus, and that is important, and we'll make mention of this toward the end of the talk. And of course, we hope that the harmless antigen is something that our immune system will handle and develop both humoral and cellular responses to, leading to immune memory. So when we think about nucleic acid-based vaccines, we're talking really about three kinds: viral vector, plasmid DNA, and mRNA. Again, all amenable to very rapid responses. And the beautiful thing is that the different vaccines can utilize the same production and purification methods. So we can scale up very, very rapidly in terms of any new threat on the globe. Once again, we still have to validate both efficacy and safety on all these platforms. So today's focus will be on mRNA vaccines. There are, the AstraZeneca and J&J vaccines are going to be viral vectored vaccines with adenovirus. But today, again, the Pfizer, Bion, and the Moderna are both conventional mRNA vaccines, and we're going to go into the differences between conventional and self-replicating or amplifying. But again, non-infectious, non-integrating, naturally degraded, egg and cell culture free, which is beautiful, and rapid and scalable production. We have some concerns, as you all are aware, about the cold chain and keeping them stable and low immunogenicity, but this really has not been shown to be a problem with the current vaccines available to us in practice. So nucleic acid vaccines, some of the beauty is that they are very amenable to customization in terms of, for example, their location. Where is the antigen going to end up? Is it going to be intracellular? Is it going to be secreted? Is it going to be membrane bound? And this determination is going to be by the signal peptide, which I'll show you in a diagram in just a second, but the signal peptide is about 15 to 30 amino acids long, and it can either be intrinsic to the natural protein sequence, but it can be engineered and direct the protein to our desired cellular compartment. Another beautiful thing in the construct is that, again, customizing, they can be targeted to MHC class 2 compartments, MHC class 1 signals, or helper T cell epitopes, which are going to amplify antigen presentation and improve our cellular immune response where immune memory is so crucially developed. Okay, so let's get down to the construct. Whether we're talking about conventional or self-replicating mRNA vaccines, it doesn't matter. Both forms are produced by enzymatic transcription, from a linearized plasmid DNA. Every part of the construct is engineered for stability, efficiency, and improved protein expression. Very importantly, the 5' cap is added to protect RNA from nuclease digestion. There are both 5' and 3' nucleases which will very rapidly destroy RNA, so the 5' cap is crucial. This is followed by an untranslated region, which is again actually engineered to increase translational efficiency. The signal peptide isn't shown here, but it would slip in right here immediately before the gene of interest, and that again is going to determine whether the location of the protein of interest is intracellular, membrane-bound, or secreted. Now, the codon sequence is important, and we're going to get into this a little bit later on, but it can be nucleoside modified or sequence optimized, and all of these are going to improve translation and productivity of the gene. And finally, there is a 3' polyadenosine tail which protects the RNA from 3' nucleases, and this is typically about 40 to 120 adenosine residues long. So this is what we're dealing with. Let's throw a signal peptide right in here. Okay, so the current COVID-19 vaccines we have, both the Pfizer and the Moderna, are both conventional or non-self-replicating mRNA vaccines. The self-amplifying or replicating, depending on whose article you read, mRNA vaccines, as we will see in a few slides, contain non-structural proteins very important for viral replication. But you have both your non-replicating, your self-replicating, and again, I'm going to get into more detail on this, but they're both inserted into lipid nanoparticles. These are targeted, these will undergo endocytosis. It's really a receptor-mediated endocytosis. And this leads to what's called a fusosome, which is the lipid nanoparticle fused with the endosome. And then there is the unloading of the payload. The mRNA must be discharged into the cytoplasm. Now, interestingly, this step, the unloading of the mRNA into the cytoplasm, constitutes the rate-limiting step in the production of the vaccine product. So in the case of the non-replicating, we will again have the mRNA uniting with the ribosomes and creating our structural protein. In the case of the self-amplifying or replicating mRNA, we get the viral replicon, which again I'll go into in just a second, and we're going to get more copies. We're going to get a higher copy number of our gene product. And in all cases, again, the signal peptide will determine, is this going to be cytoplasmic? Is this going to be transmembrane? Or is this going to be secreted? And hopefully then we're going to reach all of our professional antigen presenting cells, especially dendritic cells, and generate a very potent and protective neutralizing immune response. So regarding conventional mRNA vaccines, and again, remember, these are both of the vaccines we're currently using for COVID. Protein expression occurs very rapidly, within 12 to 24 hours after intramuscular injection, and will typically last for at least 6 days. Now, importantly, the kinetics and magnitude of the translation are influenced by many factors. The formulation itself, how it's administered, nucleoside modifications or sequence optimization. And again, I'm going to focus in a little bit on these two right here. Adjuvants can make a big difference. Now, the two vaccines we're currently using for COVID, neither of those really require adjuvant. The lipid nanoparticle itself actually has some adjuvant properties. And there can be coupling of the mRNA to small molecules, which will target toll-like receptors in certain cases, and again, increasing innate or cellular or humoral immune responses. Innate, cellular, or humoral. And that's the beautiful thing about these vaccines. So, what about nucleoside-based modification? Again, we can optimize the effectiveness of the vaccine, of the mRNA, by replacing naturally occurring nucleosides, adenine, guanine, cytosine, uracil, with modified nucleosides or by synthetic nucleoside analogs. The Pseudouridine containing mRNAs really represent the most frequent modification of mRNA. Pseudouridine is an isomer of uridine in which the uracil is attached via a carbon-carbon bond instead of a nitrogen-carbon glycosidic bond. Purification of the mRNA product is very important. Typically HPLC or FPLC is used, and these will remove double-stranded RNA contaminants. Now, again, we do want to stimulate innate humoral and cellular immunity, but too much of a good thing is too much of a good thing. And so this will reduce the stimulation of pattern recognition receptors. Again, double-stranded RNA is immunostimulatory, but we don't want too much. Excessive activation can interfere with antigen expression or adaptive immunity. So we want to get very high levels of protein production, and this has been demonstrated very clearly with many different viruses. We could throw coronavirus in here, and now we could throw humans in here as well as mice, ferrets, and non-human primates. So nucleoside-based modification is a very important customization of the mRNA vaccine. Okay, just a few words on sequence optimization. Sequence optimization, for example, a side chain modification as seen here in B or C, little side chain modification right here or right here, can skew the immune response to a specific T cell response or even skew it to a different compartment of T cells. So this could very significantly change the immune response and optimize it to get the type of cellular immune response, perhaps enhanced memory that we're looking for. Now, there are other ways to sequence optimize. One could create a chimeric peptide where, and that's shown in D here, by the way. I don't know how well you can see this, but there are slight little changes in color. So one can create chimeric peptides, and these can lead to cross-reactive T cells. This is very important because this could act against different strains of a virus. So these chimeras actually could be very crucial in our battle with COVID. We're reading about, the Kent variant, the South African variant, this variant, that variant. We're going to need to target all of these and hope that the vaccines of the day are going to cover different strains of the virus. And so this type of sequence optimization, especially chimeric peptides, may be very important for us. That's quite beautiful. So the goal with self-amplifying mRNA is to increase both the duration and the magnitude of expression, which hopefully will increase the immunogenicity of the antigen. Now, these self-amplifying mRNAs, and get ready because they're coming, and I'm going to go over that as well. These self-amplifying mRNAs typically rely upon the engineered RNA genome, of positive sense single-stranded RNA viruses, such as alpha viruses, Sinbis, Simliki-Forest, Venezuelan equine encephalitis, or flaviviruses or picornaviruses. Now, the self-amplifying mRNA genomes are typically about 10 or 11 kilobases long, and they encode 4 non-structural proteins. And together, these are called the RNA-dependent RNA polymerase complex, RDRP. Keep in mind, one of the issues or concerns with self-amplifying mRNA vaccines is, are these non-structural proteins going to interact with our immune system? This is something that we're going to need to watch, and we'll see, but they're coming. So this is going to result in the generation of many new viral genomes, up to about 2 times 10 to the 5 copies per cell. And depending upon many other conditions, this will result in a greater or lesser amount of capped mRNA encoded protein. Now, they're delivered very much the same way as conventional mRNA vaccines. They can be delivered with viral replicon particles or in lipid nanoparticles. Their expression typically will see a peak at 3 days, but these remain measurable for over 14 days, at least in non-human primates. And you saw that with our conventional mRNA, The production of the protein was typically for about 6 or 7 days. But this is very high level of correctly processed protein. And this is fortunately, again, going to be delivering antigen to a variety of professional immune antigen presenting cells, such as dendritic cells. So the activation of dendritic cells results in cytokine secretion and an adjuvant effect, which really will amplify the vaccine elicited adaptive immune response. What's beautiful is that there's a dose sparing quality or characteristic of these vaccines, and this can facilitate scale up in manufacturing. That's one of the advantages of self-amplifying mRNA over conventional. Now, the production process and stability can be a bit more challenging because they're so much larger than conventional mRNA vaccines. So let's try to summarize the advantages or disadvantages of conventional versus self-amplifying. There are indeed some advantages to self-amplifying, such as the lower effective RNA dose, the intrinsic adjuvant effect, but we have to be careful here about challenging production because these are so much longer, and there's some potential for the interaction of the non-structural proteins, the RDRP, for non-structural proteins with host factors, and these need to be addressed. We're going to find out, I think, very shortly about this. The conventional, as we're seeing, they're shorter. They're really no risk of anti-vector immunity. And we are getting protein expression, which is, in the case of COVID, certainly neutralizing and very protective. So what's on the horizon for the self-amplifying? The Imperial College London vaccine and University of Washington vaccine are both Venezuelan equine encephalitis self-amplifying. I think that the Johnson & Johnson and the AstraZeneca vaccines, which are coming, are adenovirus vector-based. So again, a lot of things which we're going to need to learn about those vaccines, I think, in the coming months. Okay, so mRNA vaccines we've seen can produce a lot of protein and really lead to a beautiful immune response, but without a protective mechanism for delivery, mRNA will be very, very rapidly degraded by 5' and 3' exonucleases. In fact, look, the size of the mRNA molecule and its negative charge prevent it from passing the cell membrane. The uptake rate for naked mRNA is estimated to be less than 1 in 10,000 molecules. That's not very good. So delivery vehicles, there are many different kinds of delivery vehicles, but lipid nanoparticles are really the ones being used at this point for the 2 vaccines we have deployed, very successfully, by the way. So the lipid nanoparticle is going to encapsulate mRNA. And again, both vaccines currently deployed are using this. And I'm going to go into this in more detail in a diagram on the next slide, but they utilize positively charged polymers to bind to mRNA, which is very negatively charged, by the way. This creates particles that behave really like viruses, shielding mRNA from the exonucleases. Now, the vehicle, again, must pass through the cell membrane. And this can be done by endocytosis or receptor-mediated endocytosis. But after uptake, the mRNA has to escape the endosome. It has to be unloaded. The payload has to be unloaded in the cytoplasm. So that's important as well. And again, as we've said, this is the rate-limiting step in delivery. So delivery system is going to affect how much gene expression we get, how much innate immune stimulation we get, and this can, again, provide, the delivery system itself can provide a synergistic adjuvant effect. Okay, a little closer look. The left panel here will illustrate a lipid nanoparticle protecting mRNA from endonucleases, 5' or 3' endonucleases. Also protecting it from phagocytosis by the reticuloendothelial system. And here you see surface ligands represented in red. I hope you can see that. which may match a specific receptor on the cell surface and facilitate receptor-mediated endocytosis. On the right is simply a more detailed diagram of the components of the lipid nanoparticle. And here you see the mRNA and ionizable lipid complexes with the negatively charged mRNA at low pH. And this is going to facilitate endocytosis and endosomal escape. Phospholipids provide structural integrity to the lipid nanoparticle, the bilayer, and support endosomal escape of mRNA into the cytosol. Cholesterol is going to stabilize the lipid nanoparticle, not only in vivo, but in the vials in which these vaccines are delivered. And there we have our old friend polyethylene glycol. Lipid-anchored PEG is going to prevent the lipid nanoparticle from aggregating and reduce non-specific interactions. So in allergy, it's very important to know the components of what we're administering to patients. And as we're going to see in a minute, it's really important to understand that probably the lipid-anchored lipid-anchored polyethylene glycol is important potentially in causing some of the systemic reactions that have been seen across the globe with the two COVID-19 vaccines. So let's talk for a few minutes about safety. Have we arrived with mRNA vaccines? Absolutely, yes. We have arrived. We are nonetheless, in an evidence-based manner, expected to address questions regarding both immediate and long-term safety. So there's so much good and positive to say about the vaccines. The manufacturing process does not require toxic chemicals or cell cultures that could contaminate the viruses. Very short manufacturing time, again, allowing for very rapid scale-up, presents very few opportunities to introduce contaminating microorganisms. No risk of infection or integration of the vector into host cell DNA. All of these things really give us a lot of peace of mind, and we can hopefully communicate these things to our patients. Are there potential safety concerns? Absolutely, yes. Of course there are. There always are. And so, however, I want to preface this section by stating that, frankly, I do not believe that any of the safety concerns justify avoiding the vaccines at this time. We all have seen the local or systemic inflammatory responses. There are some potential toxic effects of non-native nucleotides that are at least of concern, delivery system components, possibly inducing type 1 interferon responses that may be associated with inflammation and autoimmunity, some potential long-term concerns about immune enhancement, or even maybe potential cell signaling issues with spike protein. And we have now certainly heard about and seen the systemic reactions. So I'm going to focus a little bit more on these latter three concerns as we go forward with the remainder of the talk. So I'm really happy to be talking with the pediatric group. One of the reasons I joined the staff and went from practice after 30 years to an academic setting was to interact with more people. And then along came COVID. Anyway, so pediatricians are probably the group of physicians that best are familiar or most familiar with the phenomena of immune enhancement. So this is also known, or for the most part known, as antibody-dependent enhancement, where a virus may leverage antibodies or complement to amplify their replication. So RSV is probably the example best known, where in the 1960s, and actually is interesting, but this occurred in Washington, D.C. In the 1960s, there was vaccination with formalin inactivated RSV vaccine. And clearly, recipients of the vaccine, upon reinfection, were much sicker than children who had not received the vaccine. Now, the reality is there were only 2 fatalities. One is too much, but there were only 2. But 80% of children who had received the vaccine upon reinfection required hospitalization. That's pretty important. Now, the phenomena itself has been observed in vitro as well as in vivo. Certainly examples in vitro with HIV, Ebola, influenza has been seen, West Nile virus. Immune enhancement has certainly been seen, and I'm going to go over this more in detail, with both SARS-CoV and MERS coronavirus, SARS coronavirus and MERS coronavirus. And I'll go over that in detail. It's also been seen in the veterinary world with feline infectious peritonitis virus, which is a highly virulent variant of feline coronavirus that occurs in both wild and domestic cats. So cats who were immunized with feline infectious peritonitis virus had a paradoxical increase in disease severity. And this was known to be mediated by non-neutralizing monoclonal antibodies against the spike protein, even when highly diluted. The phenomena of immune enhancement may be best known in the natural world against dengue. And with dengue, those patients who have had a previous dengue infection are 15 to 80 times more likely when they suffer secondary infection to become extremely ill, very severe illness. And this is known to be mediated by the Fc portion of the antibody directed against the dengue antigen, binding to macrophages, enhancing entry and accelerating viral replication. So again, immune enhancement is very interesting and observed across the spectrum with many different viruses. We'll go into a little more detail. So this slide is very busy. Sorry about that, but it's interesting. This is a summary of SARS coronavirus and MERS coronavirus vaccine-induced enhancement in animal models. I want to stress animal models. So the reality is that depending upon the type of vaccine, you can see the many different kinds of vaccines they utilize. Inactivated virus-like particles subunit, adenovector, Venezuelan equine encephalitis virus, adenovirus, live attenuated nucleic acid, etc. Depending upon the antigen administered whole virus subunit, etc. Depending upon adjuvants, depending upon the animal species to which the vaccine was administered. There was almost every possible iteration of variable protection with some form of either increased or no lung pathology. I think that the point, oh, by the way, an interesting observation here was that when Delta inulin was used as an adjuvant, no increased enhancement was seen in terms of lung pathology. Just an interesting observation. Look, I think when you look at a slide like this, for me, this data set really just helps to assure us that the vaccine manufacturers of today had all this data and access to it, and were able to construct conditions where the likelihood of enhancement is extremely unlikely. So it's reassuring to know that this data was there, it was present, going back to attempts with SARS and MERS coronavirus to effectuate and construct effective vaccines. So I think it's good that we have the data, frankly. So again, as much as enhancement is something we should all be familiar with, and again, I think pediatricians are probably more familiar with it than other specialties, there is simply no evidence in humans that pre-existing immunity to another or even the same coronavirus will increase severity of COVID-19 disease. Human polyclonal antibodies directed against SARS can bind to COVID-19 virus and vice versa. Macaque monkeys who are infected with COVID-19 and allowed to recover were not reinfected by a second exposure to the virus. And we have all seen and read, maybe even used, convalescent plasma administering, giving high titer of COVID-19 vaccine neutralizing antibody, been shown to be effective clinically, biochemically, and radiologically, in parallel with decreasing or showing viral suppression in terms of clinical efficacy. Nonetheless, what can we say about all this in this phenomena? We can say that investigations of cross-reactivity, cross-neutralization, and cross-protection are needed. They're different phenomena: cross-reactivity, cross-neutralization, and cross-protection. And we must learn more about it. And we want to know, do pre-existing cross-reactive antibodies protect or have any detrimental effect on subsequent infection? Okay, this was an area of the literature I found quite interesting, but I had not heard a lot about it. So one of the things we talked about earlier is that when an antigen is targeted, we want to know that the antigen is harmless. So the spike protein is clearly extremely effective in terms of inducing neutralizing antibodies. Nonetheless, I did find 3 authors, Suzuki, Chen, and Patra, who raised the question as to the spike protein having very interesting properties in terms of acting as a cell signal or actually converting the ACE2 receptor into a signaling receptor. Now, the usual function of ACE2, the ACE2 receptor, is to act as a carboxypeptidase, cleaving angiotensin 2 to angiotensin and regulating blood pressure. Now, Suzuki, and this is pretty recent, 2020, treated cultured human pulmonary artery smooth muscle and endothelial cells with the S1 subunit of the spike protein. So here you see the whole spike protein, S1 and S2. And Suzuki took the S1 subunit for 10 minutes and treated these cells at a very low concentration, 130 picomolar, and demonstrated cell signaling through that receptor. Now, that is not a normal property of the ACE2 receptor. Nonetheless, Suzuki was able to confirm that. But actually, looking closer at the literature, these studies go back much further. Chen, in 2010, demonstrated that the ACE2 receptor can indeed sort of change its function from cleaving carboxypeptidase cleaving enzyme to a cell signal transducing enzyme in response to full-length spike protein. So Suzuki used the S1 subunit, Chen used the full spike protein, and both of them did demonstrate signaling through the ACE2 receptor. We'll go on a little bit more and look at this more closely. So Patra, and this is 2020, followed up on this and looked at the whole spike protein expressed by transfection in human alveolar epithelial cell lines as well as liver epithelial cell lines, and demonstrated signaling through the ACE2 receptor activating NF-kappa B, AP1, P38, and ERK mitogen activated protein kinase releasing IL-6. So again, the premise of these 3 papers is that the spike protein may, in some cases, sort of convert the ACE2 receptor into a signaling receptor, demonstrated here. So this would be the normal physiologic function of ACE2, but in certain cases, the spike protein may sort of act as a signal transducer. The ACE2 receptor can act as a signal transducer for the spike protein, causing cell signaling. Interestingly, it appears that the receptor binding domain shown here in red as part of the S1 subunit probably does not promote any cell signaling. Unfortunately, RBD-based vaccines, receptor binding domain-based vaccines, are much less immunogenic. Most of them will require an adjuvant. So they can probably convey lower potential for side effects, potentially in certain populations, but they also are much less immunogenic than the S1 subunit or the whole spike protein. So the summary of these authors was that the possibility that spike protein could trigger cell signaling events and potentially even promote pulmonary arterial hypertension or other complications. I think the operative word is in susceptible individuals will require long-term monitoring. But this is no different what the NIH has said, is that any drug released under an emergency use authorization will require long-term monitoring. And so we're all very aware of that, and I think that's what we're all doing. These patients, all patients, are being monitored very closely. Okay, we'll end up on what we're finding are the problem of systemic reactions. So first of all, systemic reactions to vaccines are extremely rare, about 1.3 per million. After the first 2 million doses of coronavirus vaccine, COVID-19 vaccine, we were seeing an incidence of about 1 in 100,000 that were systemic reactions, anaphylactic or anaphylactoid. This is not always clear. So looking at the components of the vaccine, polyethylene glycol 2000 really looks like the only excipient in the vaccine with recognized allergenic potential. Now, PEG is found, as you all know, in many products as a hydrophilic polymer. It's what's in Miralax, surfactant in injectables, vaccines, detergents, polishes, preservatives, cosmetics, many potential exposures to this. So who should we worry about potentially? Typically, if you've got a patient who is what we call a multiple drug reactor, they don't have allergy to just one drug like penicillin, but they say, I'm allergic to penicillin, I'm allergic to laxatives, I'm allergic to corticosteroids, I'm allergic to antacids. I develop symptoms immediately, itching, hives, angioedema, hypotension, shortness of breath. That patient's probably a patient we need to worry about. Do we need to worry about patients who are allergic to single foods or drugs or have insect venom hypersensitivity? No, historically, these do not predispose to allergy to other medications, drugs, or vaccines. Now, we do not currently have a skin testing protocol for polyethylene glycol. This protocol, which I have here, is from England, in which they are doing skin testing for various pegs of molecular weights. The peg in the vaccines that we currently use is peg 2000. We don't have that. We do have access to peg 3350, and we are, I think, in the process of discussing whether or not we will be able to institute a skin test protocol for polyethylene glycol. The higher the molecular weight of the polyethylene glycol, the more likely it is to induce a form of allergic reaction. Now, there is patch testing available for polyethylene glycol. However, that's really not going to help us with the acute systemic reactions. That's more for contact sensitivities and delayed type hypersensitivity, type 4 mediated gel and Coombs immune responses. I found an interesting commentary in the literature, and this was from Prié-Sella-Touré. I'm going to pronounce that wrong. But looking at the emergency medications we use containing polyethylene glycol, so a consideration whether or not it's clinically relevant, I do not know. But there are polyethylene glycol containing medications such as cetirizine tablets, loratadine, even some injectables. So to try and make sure that perhaps in the clinic, when we have our immunization clinics, that the medications we have available to treat systemic reactions really should be polyethylene glycol free, I would think, if we really think that that's what's causing our systemic reactions. So the CDC has very nicely, this was published January 6th, very recently, given us 3 groups of patients to look at: contraindications to the vaccine, precautions with the vaccine, and neither contraindication nor precaution. So in terms of contraindications to vaccination, we're looking at patients with both severe and immediate allergic reactions to any previous dose of COVID-19 vaccine or any of its components, including polyethylene glycol. Now, patients who have a known immediate reactivity to polysorbate would be of concern because polysorbate may cross-react with PEG. Interestingly, I believe polysorbate is a preservative in some of the coming DNA vaccines. These patients really should be evaluated by allergy immunology to determine whether we might or might not undertake future vaccinations with the COVID-19 vaccine. Now, as precautions, but not contraindications, we have patients who have suffered immediate allergic reactions to other vaccines or injected medications. We want to assess their risk of exposure. What is the risk of severe disease? What is their ability to be vaccinated where we can take very good care of them? And we have to consider that we might better defer on vaccination. Certainly, they should probably be evaluated by allergy. And certainly patients who have allergy to foods, pets, venom, other environmental allergens or oral medications or latex, these are not contraindications for vaccination. And by the way, the vial stoppers of these vaccines are not made with natural rubber latex. We should be observing patients closely after immunization, 30 minutes for those with a history of immediate allergic reactions of any severity to a vaccine, 15 minutes typically. Again, we want to shoot for medications that are polyethylene glycol free, I think. And we don't think that administering antihistamines prior to vaccination is recommended. We might be sort of blocking the early signs that we might see of an adverse reaction. Now, some effort should be made to identify or figure out, was this allergic? Was it an anaphylactoid, not allergic? Was it mediated by complement? So if we can draw a tryptase level after reaction within 30 minutes to 2 hours, if it's elevated, this is going to tell us that there was mast cell involvement. We can look at complement components C3A and C5A to help us determine if perhaps complement might have been activated. And we certainly want to try to determine clinically if this might have just been a vasovagal reaction. And of course, all the adverse vaccine events should be reported. So in conclusion, have we arrived? Yes. mRNA vaccines are probably our only hope to return to normal life, and the coming vaccines as well, but mRNA vaccines for now. They have all the attributes of a vaccine on demand, and again, that we can scale up rapidly. Validation, of course, of efficacy and safety must be confirmed, and there are going to be many different versions of COVID-19 vaccines. It's going to be up to you to choose which COVID-19 vaccine, mRNA, plasmid DNA, viral vector, protein-based. Novavax vaccine is protein-based. And a thorough understanding of the differences in these vaccines, how they work, and their excipients is very important to determining what patient population you're going to offer them for. We should be making an effort in the clinic to ferret out, was this allergic or was this non-allergic and perhaps an anaphylactoid by checking labs. And thank you for listening to this, and I would be happy to try and take any questions or comments.
If anybody has any questions or comments, feel free to unmute yourself or use the chat feature.
Richard, this is a very nice presentation that covers the most pertinent information a lot of us are interested in knowing. I heard that the Merck, they're not moving forward with their vaccine. I know you did a lot of research on this topic. Can you comment on, what factors played out and how they decided that their vaccine is not going to be effective? What's the difference?
Wait, I actually don't know the specific reasons why. What I do know in my reading, I've been trying to read more about, again, probably the Pfizer, of course, and the COVID, and then the Johnson & Johnson and Novavax. I have to believe it has to do with either adverse reactions, but I can't comment specifically on why Merck has abandoned that. There's a question that came in over the chat, which is, are patients on ACE inhibitors at greater risk for adverse reactions? And I am not aware of any specific publication which has pointed out that patients are more likely to suffer systemic reactions to the vaccine. And then there's another question that has come in here on the chat, which is, how does one assess immunity against COVID following either the Pfizer or Moderna vaccines? Well, I think in all cases, when we immunize patients, we can assess an antibody response, but the truth is that's only part of the immune response. When we look at an antibody titer, we can assess, did they develop antibodies, which we believe are neutralizing antibodies, but another whole aspect of immunity is, did they develop cellular immunity and immune memory? It's really not clinically available for us to assess those compartments, but we can certainly assess titers of COVID-19 IgG to assess whether or not somebody had a response, although I don't believe that that's going to be routine. And Jose Munoz just commented that a recent paper in the JAMA has confirmed that there's really no issues with the angiotensin converting enzyme inhibitors. There is a comment regarding the credit, which is the number given for the talk when entered says it's an invalid number.
I will look into that for you and see what's going on there, and if necessary, give you credit for it.
There's a question here. Have I seen patients with allergy to Miralax? Yes, actually I have. So I do think that polyethylene glycol is a potential concern, but again, going forward, we're going to see other excipients which are going to be a potential concern as well, such as the polysorbate.
Any other questions? All right, well, we will respect everybody's time and go ahead and end our session. I would like to thank Dr. Is it Loria? Loria, thank you so much for your talk today, and thanks for everybody. Evidently, Verizon's having some technical issues, so a big shout out to my coworker Melinda for helping me with this session, and Dr. Zhao and Dr. Loria for their participation as well. And we will see you guys all next week. Thank you.
Thank you.
Thanks, Kathy.
Thank you.